Hello everybody, with me Vivian Ratnawati. Here we will talk about the topic taxonomy and classifications of bacteria. Terminologies: nomenclature, concerned with assignment of name to taxonomic groups in agreement of published rules. Classification: ordering organisms on the basis of shared properties in particular taxon. Identification: process of obtaining data on the properties of organism. For example, like characterizations and determination which species is belong to based on direct comparison to known taxonomic group. Systematics: any study of nature of organism when the knowledge gained is used in taxonomy. Study of morphology, physiology and metabolic, ecology, and genetics. Pure culture: populations of individuals all derived from the same single organism. Strain: a group of pure cultures derived from a common source and thought to be the same. Species: a group of closely similar strains. Scientific nomenclature is based on a taxonomic classification system. The main aim of bacterial taxonomy is to set up classification, an ordering of bacteria into groups based on common properties. This classification can then be used to identify individual bacterial species or strains, identification, and name them. Nomenclature. Classification is used to study the relationship of different bacterial groups based on shared phenotypic properties and probably a common evolutionary history. A wide range of morphologies, biochemical, molecular characters are measured and then information is derived. From this is used to create a hierarchical classification system into which individual bacteria can be placed. Bacterial taxonomy. Carolus Linnaeus. He introduced the binomial system of nomenclature. Linnaeus established a hierarchy of taxonomic ranks. They are species, genus, family, order, class, phylum, and kingdom. Taxonomy is orderly arranging organisms under study into groups of larger units. The science of classifications and refers to classification, nomenclature for binomial nomenclature, and identification. Nomenclature of microorganisms. The rules of nomenclature: use binary names, when to capitalize, when to italicize, when to use initials, common names, subspecies and xerophores, abbreviations for species, and plural forms. The origin of names. They are descriptive, scientists' names, geographic places, and organizations. Rules of nomenclature. Binary names, invented by Linnaeus, consisting of a generic name and a species epithet, must be used for all microorganisms. Names of categories at or above the genus level may be used alone. Species and subspecies names may not, in other words, never use a species name alone. For when to capitalize, the genus name and above is always capitalized. The species name is never capitalized. For example, like Bacillus anthracis. When to italicize, names of all taxa, such as kingdoms, phyla, classes, orders, families, genera, species, and subspecies, are printed in italics and should be underlined if handwritten. Strain designations and numbers are not. If all the surrounding text is italic, then the binary name would be non-italic, Roman typeface, or underlined. Common names. Vernacular or common names should be in lowercase Roman type, non-italic, for example like Streptococcus, Brucella. However, when referring to the actual genus name or above, always capitalize and italicize. Subspecies and xerophores. For Salmonella, genus, species, and subspecies, names should be rendered in standard form. Salmonella enterica at first use. For example, like S. enterica thereafter. Salmonella enterica subspecies Arizonae at first use. S. enterica subspecies Arizonae thereafter. Names of xerophores should be in Roman type with the first letter capitalized. For example, like Salmonella enterica xerophore typimurium. After the first use, the xerophore may also be given without a species name. For example, like Salmonella xerophore typimurium. Abbreviations for species. Use SP. For a particular species, SPP. For several species, SPP stands for species plural. These abbreviations are not italicized. For example, like Clostridium SP or Clostridium SPP. Plural forms. Plural of genus is genera. Plural of species, SP, is species, SPP. Plural of medium is media. Never say this culture media. The origin of names. First, descriptive. For examples, Staphylococcus aureus, grape, like cluster of spears, golden in color. Then, Streptococcus viridans, chains of spires, green in colony color. Then, Helicobacter pylori, spiral-shaped root at the entrance to the duodenum. Second, scientist names. For examples, Escherichia coli by Theodore Escherichs. Then, Neisseria by Albert Neisser. Then, Listeria by Joseph Lister. Then, Pasturella by Louis Pasteur. Then, Yersinia by Alexander Yersin. Then, Bartonella by Alberto Barton. Geographic places. For example, like Legionella long beachii from Long Beach, California. Pasturella tularensis from Tulare County, California. Pseudomonas vermontensis from Fairmount Park, Pennsylvania. Mycobacterium genavens from Geneva, Switzerland. Blastomyces brasiliensis from Brazil. And then Providencia spp from Brown University. Organization. For example, like Legionella from American Legion. Amphibia felis from Air Force Institute of Pathology. CDC spp, Centers for Disease Control. Classification of bacteria. Berg's Manual for Classifying and Identifying Prokaryotes. They are, first, Berg's Manual of Determinative Bacteriology for morphological differential staining and biochemical tests. This one provides identification scheme for identifying bacteria and archaea. Second is Berg's Manual of Systematic Bacteriology for based on rRNA gene sequencing. This one provides phylogenetic information about bacteria and archaea. Prokaryotic systematics. Techniques for determining bacterial taxonomy and phylogeny. There are 4 classical characteristics. First, morphological characteristics. It is easy to study and analyze it. Phenotype is dependent on genotype. For example, the feature, cell shape, cell size, colony morphology, ultrastructure characteristics, staining, cilia and flagella, and motility. Second, physiological and metabolic characteristics. It is directly related to the nature and activity of microbial enzymes and transport proteins. For example, carbon and nitrogen sources, cell wall constituents, energy sources, fermentation products, general nutritional type, growth temperature optimum and range, mechanism of energy, osmotic tolerance, oxygen relationships, secondary metabolism formed. And the last, mechanism, motility. Third, kinetic characteristics. It studies of chromosomal gene exchange through transformations, conjugations, and transduction. Thus, ecological characteristic, ability of a microorganism to colonize a specific environment. Taxonomically important ecological properties are life cycle patterns, the nature of symbiotic relationships, and the ability to cause disease in a particular host. Techniques for determining bacterial taxonomy and phylogeny. There are 4 molecular characteristics. First, nucleic acid-base compositions. For example, Kc ratio. Second, nucleic acid hybridizations. For example, DNA homology, DNA base sequence, and RNA sequence. Third, nucleic acid sequencing. For example, rRNA from small ribosomal subunit 16S and 18S rRNA from prokaryotes and eukaryotes respectively. Last, genomic fingerprinting. For example, DNA fingerprints or RFLP, restrictions fragments length polymorphism pattern. Polyphasic taxonomy. First, phenetic classification. Classical microbial taxonomists relied exclusively on a phenetic system, which organized organisms according to mutual similarity of their phenotypic characteristics. Second, phylogenetic classification. Phylogenetic or phyletic classification systems sought to compare organisms on the basis of evolutionary relationships as srRNA sequencing. Third, genotype classifications. Genotypic classification seeks to compare the genetic similarity between organisms. Individual genes or whole genomes can be compared. Berger's Manual of Systematic Bacteriology. First edition, published in 4 volumes, mainly based on phenotypic characters. Second edition, published in 5 volumes.